The human body manages its energy demands through a chemical binary, storing energy when resources are abundant and breaking it down during a deficit. Within lipid metabolism, these two physiological states are governed by specific pathways: fatty acid synthesis and fatty acid oxidation. These pathways are structured as biochemical opposites. If the cell allowed both systems to run in the same space at the same time, they would cancel each other out. To prevent this, the body physically separates the two processes. Oxidation, the breakdown process, is confined to the mitochondria. Synthesis, the building process, occurs in the surrounding cytoplasm. This spatial isolation prevents a futile cycle, where the cell would waste energy building and destroying fat simultaneously. The mitochondria serve as the primary site for fatty acid oxidation, functioning as the hub for energy release. The mechanism driving this breakdown is a cycle known as beta oxidation. Beta oxidation operates as a repeating loop that sequentially cleaves two carbon units from the end of a long fatty acid chain. Each turn of this cycle releases a specific two-carbon product, acetyl-CoA. These acetyl-CoA molecules are then fed directly into the cellular TCA cycle within the mitochondrial matrix. The TCA cycle processes these units to generate high-energy electron carriers, specifically NADH and FADH2. These carriers then proceed to the electron transport chain, providing the necessary substrate to produce high yields of ATP. This makes fatty acid oxidation a strictly catabolic process, deployed only when the body's systemic energy demands require it. This energy release depends on access to the mitochondrial machinery, which is protected by the highly selective inner mitochondrial membrane. Long-chain fatty acids in the cytoplasm are unable to cross this barrier independently and are repelled by the membrane. To enter the mitochondria, the cell must employ a specialized biochemical transport system, the Carnitine Shuttle. The cell attaches a carnitine molecule to the activated fatty acid, granting it the chemical identity required to pass through the membrane. This new complex is then actively moved across the barrier and into the mitochondrial matrix for processing. Because this shuttle regulates access to the oxidation machinery, it dictates whether fat can be burned at all. Without carnitine, beta oxidation stops. Crossing back into the cytoplasm brings us to the domain of anabolic construction and energy storage. Here, acetyl-CoA is not destroyed for energy, but is instead used as the universal building block for constructing new fats. The entire synthesis pathway is controlled by a rate-limiting enzyme, Acetyl-CoA Carboxylase, or ACC. ACC catalyzes the conversion of acetyl-CoA into melanyl-CoA, providing the direct precursor needed to elongate the fatty acid chain. The chain then grows step by step through a recannative enzymatic process that adds two carbons at a time. Unlike the breakdown of fat, this synthesis is energy-intensive, actively consuming cellular ATP to build these molecular bonds. Synthesis also requires a high volume of reducing power, which is provided specifically by the carrier NADPH. This process represents a long-term energy investment, used by the cell to store surplus resources for future use. Examining these pathways side by side reveals how the chemical steps of degradation and synthesis are structured. Looking at the left column of this chart, beta oxidation follows a sequence of oxidation, followed by hydration, and a final oxidation. The synthesis process on the right is the reverse. It requires reduction, followed by dehydration, and a final reduction. They use distinct carriers as well. Oxidation generates NADH and FADH2, while synthesis consumes NADPH. This chemical divide matches the physical one. Breakdown is locked in the mitochondria, while assembly is restricted to the cytoplasm. Because these pathways utilize opposing chemical sequences, the mechanics of one provide a functional blueprint for the other. These molecular pathways are governed by systemic hormonal signals that respond to the body's overall state. This table shows that in the fed state, high blood glucose triggers the release of insulin from pancreatic beta cells, impacting the storage of fat and proteins. Insulin acts as a molecular switch, upregulating acetyl-CoA carboxylase to prioritize the synthesis of new fatty acids. Conversely, during fasting, the body releases counter-regulatory hormones like glucagon and epinephrine to mobilize stored energy. These hormones activate hormone-sensitive lipase to release stored FODs, forcing them into the mitochondria for beta oxidation. This creates a coordinated survival system. While the liver recycles lactate from muscles through the Cori cycle to maintain blood sugar, it uses the energy harvested from fatty acid oxidation to power the process. Looking at the summaries for blue-coded oxidation and orange-coded synthesis, the relationships are clear. One pathway releases energy in the mitochondria, while the other stores it in the cytoplasm. Lipid metabolism is a binary system governed by hormones that shift cellular resources between long-term storage and immediate metabolic survival.